Hello everyone. So last meeting we're able to discuss about carbohydrate metabolism, the different processes that were involved, particularly involving glucose and glycogen. But we have our primary focus in glucose metabolism since glycogen serves as backup source of glucose. So glycolysis product was pyruvate. And if you recall, one molecule of glucose yields two molecules of pyruvate. Okay, so meaning to say, since there are two molecules of pyruvate, so there are two precursors or two molecules that serve as precursor for acetyl coenzyme A. So acetyl coenzyme A is the oxidized form of pyruvate. Take note, glycolysis or gluconeogenesis occurs in the cytoplasm of cells. Now, for our discussion today, we'll talk about Krebs cycle or the citric acid cycle or tricarboxylic acid cycle. So, Krebs cycle is the primary metabolic pathway through which aerobic energy, meaning to say you're requiring oxygen or there is oxidation involved, it's released from the carbs, proteins, and fats in usable form. Okay, meaning to say there is ATP produced. So it's called Krebs cycle because of its proponent Hans Krebs, who won the 1953 Nobel Prize in Medicine. This cycle occurs in the mitochondria and it has a series of 8 irreversible steps. If you recall, in glucose metabolism, so the four processes tend to be reversible. However, for Krebs cycle, its intermediary forms may be reversible, but the whole cycle is not. Meaning to say, there's only one way for the cycle to proceed. Okay. Now, prior to the Krebs cycle, so from the cytoplasm, pyruvate is transported into the mitochondria. However, it is further oxidized to form acetyl coenzyme A. Okay, so a byproduct for oxidation of pyruvate is carbon dioxide. And the enzyme responsible for transforming pyruvate into acetyl coenzyme A is your pyruvate dehydrogenase. So do take note the patterns of how enzymes are named. So oxidation gani, so expect usually in metabolic context, metabolic pathway, oxidation is merely dehydrogenation. And one of the products of oxidation or metabolic oxidation is carbon dioxide. Okay. So our coenzyme, another coenzyme involved is NAD. So if you still remember in the first discussion on metabolism, so the two important cofactor or coenzyme are your NAD and FAD. Okay. But for this cycle, Krebs cycle, the most important coenzyme that will bind to the derivative of pyruvate is coenzyme A. Okay. So acetyl coenzyme A is then the first molecule that's the start of Krebs cycle. This is how the cycle is visualized or is represented. Okay. So if you recall, there are two pyruvate molecules that are products of one glucose molecule that is undergoing glycolysis, meaning to say there will be two acetyl coenzyme A as well for every molecule of glucose. Okay? So 1:2 ilang ratio. One glucose molecule, two pyruvate molecules, two acetyl coenzyme A. Because there will be two pyruvate molecules that will be oxidized, there will be two acetyl coenzyme A that will be produced. Okay. Then this acetyl coenzyme A or acetyl CoA is combined with oxalate or oxaloacetate. So acetyl-CoA and oxaloacetate will combine with each other and a new compound is synthesized. That is your citrate. Now, the enzyme responsible for catalyzing the formation of citrate is your citrate synthase. Now, take note, when you encounter enzymes such as synthase or can also encounter enzymes that are named synthetase, these two enzymes catalyze the same reaction, that is synthesis. Synthesis of new compounds or molecules, but the major difference is that for synthase, you require no energy. For synthetase, you will require energy. Okay? That's the major difference. Again, synthase, synthesis, no energy. Synthetase, still synthesis but requires energy in the form of ATP or its equivalent GTP. Okay? Step 1 is this one. Okay? Oxaloacetate plus acetyl coenzyme A to form citrate. Step 2, you will transform citrate into its isomer. So, the enzyme involved is aconitase. Okay? Meaning to say, aconitase is an enzyme that forms isomers, isomerase. Okay? Now, from there, we move to step 3. Step 3, okay. Step 3 is from isocitrate, you form alpha-ketoglutarate. Now, this is the rate-limiting step because it predicts or determines how fast the rest of the cycle will proceed. And it will depend on the level of ATP present. If there is a high level of ATP, then TCA will proceed slower. Otherwise, if there is low ATP level in the cell, then it will go faster. And this rate limiting step is like a controller. So if merry-go-round pa ni siya, so kani siya ang nag-control or nag-maneho sa whole nga cycle. Okay? So whole nga ride. If this is the merry-go-round kani, then kani siya ang station. Okay? And alpha-ketoglutarate is also one of the pathways towards protein metabolism. Okay? There is a passageway or naay in entry point si protein metabolism kani nga step. Okay? Now, the byproduct is carbon dioxide. And the enzyme responsible to catalyze this reaction would be your isocitrate dehydrogenase. Meaning to say, isocitrate is oxidized to form alpha-ketoglutarate. Okay? And if you notice, the same cofactor is present, such as that in pyruvate oxidation, you have NAD. And same byproduct, carbon dioxide. In other words, byproducts or energy which will not be successfully converted into usable form such as ATP will either be emitted as carbon dioxide or heat. Okay, duharan na. Carbon dioxide or heat. Excess heat. Ang main energy form nga ma-produce is, of course, ATP. Then, step 4, alpha-ketoglutarate is further oxidized to form succinyl-CoA. Okay, so enzyme is alpha-ketoglutarate dehydrogenase. Then, the next step, step 5, from succinyl-CoA, you form succinate. And if you notice, it requires GTP or GDP, which is an equivalent of ATP. And it is, meaning to say it is requiring energy in the form of phosphate. So there is energy involved to form succinyl coenzyme A into succinate. Meaning to say, succinate thiokinase is also called succinate synthetase because you are synthesizing succinate. This is succinate synthetase. Okay. So this is just the sibling of citrate synthase, no? You have synthase sa taas, you have synthetase here, or thiokinase. That is step, so 1, 2, 3, 4, this is step 5. So tulo na lang. Step 6, oxidation again, but this time, cofactor involved is FAD. Okay? Then, step 7, you have a hydrase. Meaning to say, you added water molecule to fumarate to form malate. And the enzyme involved is fumarate hydrase, or other terms or other references will also call it fumarase. Okay? So, fumarase is simply hydrase. So, opposite siya ni hydrolase, no? Because hydrolase, you are breaking water molecule. But for hydrase, you are adding the water molecule. Okay? Then, step 7. Okay, muna siya step 7. Then steps 8. Step 8 is the last one. From malate, it is oxidized to form oxaloacetate. And again, that means one acetyl coenzyme A has been consumed. Then since duha man ang mo enter, so kani nga oxaloacetate will then combine with the other acetyl coenzyme A produced from one molecule of glucose. Okay? And then the cycle will repeat. So that means there are two acetyl coenzyme A for one molecule of glucose. So for every molecule of glucose, there are 2 rounds of Krebs cycle. Because there are 2 pyruvate molecules that are produced and it is oxidized, so there will be 2 acetyl coenzyme A is formed. Okay? So, also, the total generated ATP will be 12. So, based on the summary here, so there will be 12 ATP that will be generated. Since there are 3, so if you count, there are 1, 2, 3 NADH produced, that is equivalent to 9 ATP. There is 1 FADH2 produced, which is equivalent to 2 ATP. And you have 1 GTP produced, 1 ATP, since 1 GTP is to 1 ATP. Adding, so nakay 12 ka ATP for every molecule of acetyl coenzyme A. So, meaning to say, for 1 molecule of glucose, duha man ka acetyl coenzyme A, so ma-times 2 pod ni siya. Okay? If duha na ni. Okay? So, duha, ma-times 2 pod ni siya. Okay? So, that's for Krebs cycle. Okay, so let's try to sum up. Okay, so we have there rate limiting step. Okay, so here is another diagram to show or that emphasizes. So ganinang emphasis was on the enzymes and the products or the names of the products. This is showing us the number of carbon molecules. So this is sama ni nga molecule class. Okay, you guessed it right. There are 3 carbons, so that's pyruvate. Then, oxidation, meaning to say this is acetyl coenzyme A. Diba? This one here, there are 4. So, this is oxaloacetate. Then, kani siya, mumukombine ni, you have 4 plus 2, 6. This is your citrate. Okay? Kani, pyruvate. This one, oxaloacetate. And then, the 2 carbons, you have acetyl coenzyme A. And the rest are the intermediates. Kani, isocitrate. Then, the rest. Okay? So, let's try to answer. You may pause the video and then you may resume if you already are confident of the answer. Okay? So, let's try to answer, assuming you've already paused the video. Okay, so for pyruvate oxidation, so obviously, the first molecule is pyruvate. Okay, so pyruvate enters the mitochondrion from the cytoplasm. So, siya ang product of glycolysis. Okay? So, how many atoms are removed? Ingon siya, 1 atom is removed via blank. And blank is removed using blank. Okay? So, we can refer to the diagram earlier. So, how many atoms are removed? Okay? So, earlier you have 3. Yeah, karon duha, meaning to say carbon ni siya. This is carbon. Carbon via, so that's it is referring to a process. Meaning to say, since it loses carbon, so we are pyruvate undergoes a process called, so it's actually called decarboxylation or decarboxylation kay gitangtang si carbon. And aside from carbon, unsa pa may nawala? So if you notice here, CNAD plus nga cofactor, ni-gain siya hydrogen. Meaning to say, pyruvate also lost a hydrogen molecule. So here, hydrogen is removed. So hydrogen is removed using the cofactor or coenzyme NAD, which is in its reduced form. Now, then, you have already lost carbon. You also lost hydrogen. So, pyruvate lost carbon from 3 na himo lang duha. Then, something daw is attached to the remaining blank atoms. So, meaning to say this is also carbon. Creating blank, which then enters the Krebs cycle. So, we know that the precursor is pyruvate, and once it is oxidized, the product is acetyl coenzyme A. Okay? This is acetyl coenzyme A. Kani. So, what was attached to the remaining carbon atoms? Okay? So, that means coenzyme A was attached to the remaining carbon atoms. Okay? So, that paragraph refers to pyruvate oxidation. Okay? Now this time, let's move to the Krebs cycle proper. Okay? So our product here was acetyl coenzyme A. Meaning to say that will enter. So this one here, ang first is acetyl coenzyme A. Enters the cycle and then combines with blank to make the 6-carbon compound blank. We know that acetyl coenzyme A has 2 carbons here. And if we look at back, meaning to say, acetyl coenzyme A combines with oxaloacetate to form 6-carbon compound citrate. Okay? So this is oxaloacetate to form the 6-carbon compound which takes the name of our cycle, citrate. During the blank steps of the Krebs cycle, how many steps naa sa Krebs cycle? So there are 8. This is during the 8 steps of the Krebs cycle. Blank undergoes a number of reactions, releasing blank and blank in a number of steps. So our primary intermediate is citrate. So same ra gihapon. Okay? And it undergoes a number of reactions. What are released? So usable form of energy and waste product. What is our waste product? So we have carbon dioxide, and usable form, you have the energy currency ATP in a number of steps. And then blank is eventually converted into blank, so it can be used again during the Krebs cycle. What is the compound that is reusable? Okay, so meaning to say, when we look at the diagram, asa ani ang ma-reuse? Okay, so nara gyud na sa tumoy nga mga products. We don't reuse acetyl coenzyme A because it is only one time, and the reason there is a repetition of the cycle, it's because there are 2 acetyl coenzyme A that enters. Meaning to say, ang ma-release is ma-reuse rather is oxaloacetate. So unsay intermediate before major intermediate before oxaloacetate? That is citrate. So meaning to say, citrate is eventually converted into oxaloacetate, so it can be used again during the Krebs cycle. Okay? So that's for Krebs cycle. Now, if we go to the products, so what is released as waste can either be heat, but since we're concerned of molecules, so we use carbon dioxide is released as waste. Now, what are the other products of Krebs cycle DI? If we look at apart from citrate, apart from oxaloacetate, apart from ATP rather, so looking at here, we have ATP, GTP in here. Other products have carbon dioxide. Carbon dioxide, we also have NADH and FADH2. So meaning to say, other products apart from ATP and carbon dioxide are your cofactors. We have NADH and FADH2. These are the reduced forms. Okay? They move to the next stage of cellular respiration. Energy is released in the form of currency ATP. A glucose molecule produces blank molecules of blank because 2 molecules of blank are created from each molecule of blank. So each molecule of kani glucose. Then 2 molecules of what are created from each molecule of glucose. So from glycolysis, you have glucose to pyruvate. So meaning to say, this is pyruvate. And pyruvate is the precursor for acetyl coenzyme A, 'di ba? And then acetyl coenzyme A is converted to energy. But what is the form of energy? So energy is released in the form of ATP. Pila ka ATP ang ma-produce per glucose molecule. Okay? If we go back, it is stated here that for every acetyl coenzyme A molecule, you have 12 ATP. But then there are 2 that enters, meaning to say there are 24. Because for 1 glucose, 2 pyruvate, 2 acetyl-CoA, 24 ATP. So, meaning to say, 1 glucose molecule, 2 pyruvate, 2 acetyl coenzyme A, 24 molecules ATP. Okay, so those are the products of Krebs cycle. Okay, so that's for Krebs cycle. I hope you get the gist of the whole cycle, and I'll see you in the next video.